A man we'll call David Cohen opens an email from a genetic testing company he'd almost forgotten he used. The subject line says he has a new match. He clicks through, expecting a distant cousin from Poland, maybe, where his father's family had lived before the war. Instead, the report shows a near-perfect match on his Y chromosome, the strand of DNA passed from father to son, virtually unchanged for generation after generation, to a man living in Morocco. Different country, different language, a surname that has nothing to do with Cohen. No known family connection of any kind, no shared great-grandparent, no overlapping branch on either man's family tree going back as far as either of them can trace it by paper alone. David reads the explanation twice, then a third time, the way people do when a result seems to be telling them something slightly larger than they were prepared for. The shared signature is called the Cohen modal haplotype. It's the genetic marker researchers first identified in 1997 among men who traced their family line to the Cohanim, the Jewish priestly caste. Men whose tradition holds that they descend, father to son, from a single ancestor who lived over 3,000 years ago in the ancient Near East. David's own family always said they were Cohanim. He hadn't thought much of it. It was the kind of thing you'd hear from a grandfather at a holiday table, filed somewhere between family history and family folklore, repeated more out of habit than conviction. Now, there's a stranger's name sitting in his inbox, a man from a different continent, carrying what looks like the same piece of DNA, and no easy explanation for why, beyond the one the science is about to offer. That's the story this piece is actually about, not David, specifically, since his case is one illustration among thousands of similar results returned every year by testing companies processing samples from around the world. But the real peer-reviewed science behind why a moment like this happens at all, and what it can and can't actually tell someone who receives it. Over the past 25 years, geneticists have compared Jewish populations from nearly every continent on Earth against reference populations from Europe, the Middle East, Africa, and Asia, using genome-wide data, Y chromosome analysis, and disease gene mapping. The findings, published in Nature, the Proceedings of the National Academy of Sciences, and the American Journal of Human Genetics, turned out to be more precise and more carefully qualified than most coverage of this research has managed to convey. This is that research, and it's also, in a real sense, David's story, because it's exactly what shows up when a family tradition, carried for generations without paperwork, finally meets a laboratory equipped to test it. One clarification first, because it shapes everything that follows, and because it's the difference between good science journalism and the kind of headline that misleads before the first fact even arrives. Modern population genetics does not sort humanity into races. Researchers studying groups like Jewish diaspora communities are not measuring anything resembling racial categories, and no serious paper in this field frames its findings that way. They're tracing ancestry, specific testable lines of descent, and intermarriage, measured through actual shared DNA rather than through appearance, geography, or any broader proxy category. That distinction is what let researchers ask a genuinely precise question about people like David. After 2,000 years of dispersal, would Jewish communities scattered from Yemen to Poland to Ethiopia still carry a detectable, shared signature pointing back to a common origin? Or would 2 millennia of separation, intermarriage, and life among dozens of different host populations have erased it, leaving only the genetic fingerprint of wherever each community happened to settle? Let's start where David's own result actually sits, inside the most specific and, honestly, most remarkable branch of this research, the genetics of the Cohanim themselves. In 1997, researchers Carl Skorecki and Michael Hammer set out to test something that sounded on paper almost too neat to be real. If Jewish oral tradition was right, if men identifying as Cohanim really did descend patrilineally from a single ancestor, transmitted by religious practice rather than written record for 3,000 years, that lineage should, in theory, leave a detectable trace on the Y chromosome, the one part of the genome that passes from father to son with almost no mixing along the way. They compared Y chromosome markers among self-identified Cohanim against other Jewish men, publishing the result in Nature. A distinctly high proportion of Cohanim, sampled independently across both Ashkenazi and non-Ashkenazi communities, men with centuries of separate history, no contact with each other, sometimes speaking different languages entirely, living continents apart, shared the same Y chromosome signature. Researchers named it the Cohen modal haplotype. It made headlines well beyond the scientific press precisely because it did something genetics research rarely manages. It took a specific religious tradition, stated in specific terms, and returned a specific, testable answer. 12 years later, a follow-up study using a far larger set of genetic markers went back and tested the finding more rigorously, because that's what good science does with an exciting early result. It doesn't just repeat it and move on. It tries to break it, sharpen it, or find the parts of it that were too simple the first time around. The picture that came back was more precise and slightly more complicated. The refined version of the Cohen modal haplotype accounted for close to 30% of Cohanim Y chromosomes across both Ashkenazi and non-Ashkenazi communities, was essentially absent in non-Jewish comparison populations, and traced to a common ancestor estimated at roughly 3,200 years old, a date broadly consistent with the traditional timeline, though not proof of it. But the same study identified additional, separate paternal lineages among other self-identified Cohanim, including a second lineage found at around 14% frequency, estimated at roughly 4,200 years old. The honest conclusion wasn't one founding father. It was a small number of distinct paternal lines, established before the communities carrying them scattered into separate diaspora populations. Later commentary on the research has been direct about this. The fuller genetic record doesn't support a single founder for all Cohanim. It supports something closer to a small founding cohort. David's match statistically likely sits inside that broader, more complicated picture. A real, deep, ancient paternal connection, just not quite the single unbroken line the simplest version of the story suggests. When he mentioned this result to his father didn't seem surprised by the complexity at all. He just said, "Of course, there's more than one of us. There were a lot of priests." It's the kind of answer that sounds almost dismissive, until you realize it's actually the more sophisticated position. An old man who'd never read a population genetics paper in his life, arriving, by instinct and family memory alone, at roughly the same conclusion the 2009 study spent years of careful sequencing to confirm. Zoom out from David's family, and the same basic question: does a shared identity carried for 2,000 years across separate continents still show up in DNA? Has been tested at a much larger scale, using entire genomes rather than a single chromosome. The results are direct. Most Jewish diaspora communities tested, separated for centuries by geography, language, and independent political history, cluster together genetically, more closely with each other than with the non-Jewish populations they lived among for generations. The data is consistent with most contemporary Jewish communities, descending substantially from a shared Middle Eastern ancestral population, with each community picking up its own additional local genetic influence from its host society, layered on top of that shared foundation over the centuries that followed. A Jewish family that spent 800 years in Poland and a Jewish family that spent 800 years in Yemen still carry more in common with each other, genetically, than either carries with the non-Jewish neighbors who lived next door to them the entire time. That finding came from a 2010 study in Nature, led by geneticist Doron Bahar, working with researchers across more than a dozen institutions on 3 continents. The team compared genome-wide data from 14 Jewish diaspora communities, Ashkenazi, Sephardic, and Mizrahi populations from Iraq, Iran, Syria, Yemen, and elsewhere, along with smaller groups like Georgian and Kurdish Jewish communities, against 69 non-Jewish populations across the Old World, 25 of which had never appeared in a comparative genetics study before. It remains one of the broadest studies of its kind, and its scale is part of why its conclusions carried as much weight as they did within the field. Ashkenazi, Sephardic, and Mizrahi participants form genetically distinguishable but closely related clusters positioned closer to each other and to non-Jewish Middle Eastern populations than to the European populations many of these same communities had lived among for over 1,000 years. The researchers also noticed something easy to miss. Caucasus and Middle Eastern Jewish subgroups showed more genetic exchange with each other specifically than either group showed with its own local non-Jewish neighbors, more consistent with ongoing contact between dispersed Jewish communities themselves, maintained across enormous distances, than with each community simply dissolving into whatever society surrounded it. That finding mattered for a specific, long-running historical argument, one worth pausing on, because it's the kind of claim that circulates widely in popular discussion and rarely gets tested directly against actual genetic data. For decades, a theory known as the Khazar hypothesis held that Ashkenazi Jewish ancestry derived substantially from the Khazars, a Turkic-speaking people who ruled a kingdom in the Caucasus and converted to Judaism sometime around the 8th or 9th century, before that kingdom eventually collapsed a few centuries later. The theory has circulated in various forms since the 19th century, and it makes a specific, testable prediction. If true, Ashkenazi participants should show a meaningfully stronger genetic connection to populations from the Caucasus region than to populations from the Middle East. The Behar study tested this directly. So did a dedicated 2013 follow-up, built specifically to check it, using an expanded set of 261 samples from 15 populations across the Caucasus and the region just north of it, populations never previously compared against Ashkenazi Jewish samples at this scale, and chosen specifically because they represent the closest available genetic proxies for the historical Khazar population. Neither study found the signal the hypothesis predicts. Ashkenazi participants showed no particular genetic affinity with Caucasus populations, including the ones considered most likely to represent Khazar descendants. Their closest relationships remained with other Jewish communities, and after that, with non-Jewish populations from Europe and the Middle East, a pattern far more consistent with substantial Middle Eastern origin, followed by centuries in Europe, than with a Caucasus conversion event as a primary source of ancestry. It's a clean example of a specific popular historical claim meeting a specific, well-designed genetic test, and simply not surviving contact with the data. Genome-wide comparisons like these are powerful, but they blend the inheritance of both parents together, generation after generation, diluting the signal each time a new marriage brings a new, unrelated set of ancestors into the mix. The Y chromosome, the tool that produced David's own result, works differently. It passes from father to son almost unchanged, without the reshuffling that happens elsewhere in the genome through recombination. Functioning as a direct paternal line timestamp, researchers can trace back through centuries, sometimes millennia, with unusual clarity. It's a narrower lens than a full genome comparison, following only one line among the thousands of ancestors any person actually has, but it's also a sharper one, precisely because nothing dilutes it along the way. The 2000 study in the Proceedings of the National Academy of Sciences used exactly this tool at a much wider scale, analyzing Y chromosome markers from 1,971 men across 29 populations worldwide, Jewish communities alongside non-Jewish populations from the Middle East, North Africa, Sub-Saharan Africa, and Europe, one of the broadest comparisons of its kind attempted up to that point. The team examined a set of biallelic genetic markers, small stable variations in the DNA sequence that change only rarely across generations, making them especially reliable for tracing deep ancestral relationships rather than recent family history. The result: Jewish men across multiple geographically separated communities shared a common Y chromosome signature, specifically with non-Jewish populations from the Middle East, including Palestinians, Syrians, and Lebanese populations, diverging significantly from the patterns found outside that regional cluster, including the European populations several of these same Jewish communities had lived among for the better part of a millennium. The researchers' interpretation was direct. Widespread intermixing with host populations, sustained over many centuries, would have been expected to erode that shared signature into something closer to each community's local surroundings, the way a river gradually smooths a stone until its original shape disappears. That erosion isn't what the data showed. A community that spent centuries in Yemen and a community that spent centuries in Central Europe, with essentially no contact between them, still carried Y chromosome signatures closer to each other than to the populations immediately around them. A follow-up study the next year, led by geneticist Almut Nebel and published in the American Journal of Human Genetics, sharpened the picture further, comparing Jewish Y chromosome data directly against Palestinian Muslim Arab, Bedouin, and Muslim Kurdish populations. 526 Y chromosomes across 6 Middle Eastern populations in total. Roughly 70% of Jewish Y chromosomes and roughly 82% of Palestinian Muslim Arab Y chromosomes belonged to the same core chromosomal pool, a pattern the researchers described as a remarkable degree of genetic continuity in both populations, despite Jewish communities' long separation and wide dispersal. Kurdish and Sephardic Jewish participants turned out to be genetically indistinguishable from each other on this measure. Ashkenazi participants showed a small, statistically real divergence from both, attributed most plausibly to limited gene flow from European populations or to genetic drift during centuries of relative isolation, rather than a fundamentally separate origin. Later work, adding Druze populations into the comparison, found the same pattern again from a different angle. Palestinian and Druze populations, both showing clear genetic proximity to Ashkenazi and Iraqi Jewish populations specifically, two more independent data points converging on the same underlying story. This is where the human cost of this history becomes unavoidable, and worth naming directly, because the genetics only makes sense once the history behind it is on the table. A history of expulsion, forced relocation, and, in later centuries, organized violence, all of it now legible in a very specific and testable way in the genome of the population that survived it. Consider a woman we'll call Ruth, a distant cousin in David's extended family, who went through genetic screening before starting a family of her own. She'd expected the appointment to be routine, a formality, something her doctor's office recommended for everyone in her position. Her results came back flagging something specific to her ancestry, an elevated carrier risk for several inherited conditions, including Tay-Sachs disease and mutations in the BRCA1 and BRCA2 genes tied to breast and ovarian cancer. Her doctor explained why, almost apologetically, sensing she hadn't expected the conversation to go this direction. These aren't random findings scattered across the population. They trace to a documented population bottleneck, a period when the reproductively active Ashkenazi Jewish population contracted sharply, likely sometime in the latter half of the Middle Ages, before expanding again from a startlingly small founding group. Geneticist Shai Carmi of the Hebrew University of Jerusalem places that effective founding population in the hundreds to low thousands, a striking number given that the global Ashkenazi population today numbers in the millions, nearly all descended from that same narrow group within the last 7 or 8 centuries. A widely cited 2004 analysis, using a statistical test specifically built to distinguish a genuine founder effect from other explanations like natural selection, places the sharpest such contraction between roughly 1100 and 1400 CE, with earlier genetic evidence pointing to a separate bottleneck around the year 75 CE, at the start of the wider diaspora following the destruction of the Second Temple in Jerusalem. When a population passes through a bottleneck that narrows and then rebuilds itself while marrying largely within the group, a pattern reinforced by both religious practice and, for long stretches of medieval and early modern European history, by external exclusion that made marrying outside the community difficult or dangerous, rare variants carried by the surviving founders end up disproportionately represented in every generation that follows. A mutation that might have stayed rare, diluted across a large open population, instead gets concentrated, generation after generation, inside a group that kept marrying largely within itself. This is the documented origin of the roughly 200 known founder mutations now cataloged in Ashkenazi Jewish populations. It's also, not incidentally, one of the field's clearest translations from population history into direct practical medicine. Since large-scale carrier screening began in the 1970s, Tay-Sachs disease has been reduced by more than 90% in screened Ashkenazi populations, one of the more striking public health success stories in modern genetic medicine, built entirely on understanding exactly the kind of population history this research maps. Ruth's own screening result, unsettling as it was to receive in the moment, exists because of exactly this research. And it's part of the reason her own children, if she has them, are far less likely to inherit a condition that would have gone undetected in her grandmother's generation. Not every part of this story resolves as cleanly, and it's worth being direct about where it doesn't, because pretending otherwise would misrepresent research that has, at every stage, been careful to say so itself, rather than smoothing over the parts that complicate a tidier narrative. The Bahar Study's broader survey included the Bene Israel community of India, and found that population substantially admixed, carrying a real, detectable Jewish ancestral signal alongside a very significant, clearly identifiable component of South Asian ancestry, reflecting centuries of local intermarriage in a community whose own oral tradition places its arrival on the Indian subcontinent many centuries ago, long before most other Jewish diaspora communities had settled into the regions they're associated with today. Later research on this specific community described its members as carrying substantial ancestry from both Jewish and Indian populations, each contributing a genuinely large share rather than one being a minor addition to the other, a genuine, well-documented blend rather than a single clean thread. Ethiopian Jewish communities show a related pattern: substantial regional East African ancestry alongside a smaller, more limited signal shared with other global Jewish populations, consistent with many centuries of largely separate development in the Horn of Africa before renewed contact with other Jewish communities resumed in the 20th century, much of it only within living memory for many families involved. These aren't exceptions that undercut the research. They're what an honest survey should produce: a range of outcomes reflecting each community's genuinely distinct history, rather than one identical story stretched uniformly across groups separated by thousands of miles and, in some cases, well over 1,000 years of independent history. A study that returned the exact same result for a community that spent 1,000 years in Lithuania and a community that spent 13 centuries in India would be far more suspicious, methodologically, than one that found real, well-explained variation between the two. The differences are, in their own way, part of what makes the consistent signal found across so many of the other communities so striking by comparison, a signal that held up again and again in populations with no reason to resemble each other except the one thing the DNA kept pointing back to, which brings the story back to David, because that's where it actually started and where it's worth ending too. Weeks after the original match appeared in his inbox, David wrote to the stranger in Morocco. It took him a few days to work up to it. There's something strange about introducing yourself to someone on the basis of a shared chromosome and nothing else, no shared language guaranteed, no certainty the other person will even want to talk. They exchanged a few messages anyway. Polite, curious, a little formal. The way two people are when the only thing they're certain connects them is a molecule neither of them fully understands the mechanics of. The Moroccan man's family, it turned out, also carried an oral tradition of priestly descent, passed down the same way David's had been. Not through paperwork, not through any surviving document either family could point to, but through what a grandfather told a father and a father told a son, generation folding into generation with nothing written down at any point along the way. Neither man could say, with any certainty, exactly how their two family lines had diverged, or when, or under what circumstances, one branch had ended up in Eastern Europe and the other in North Africa. The genetic evidence placed their shared ancestor somewhere in the range of 3,000 years back, give or take a few centuries, long before either of their families' more recent histories in Poland or Morocco had even begun, long before either country existed in anything like its current form. David has said since that the strangest part wasn't the science itself. It was realizing that a family story he'd always treated as something closer to folklore. A nice thing, his grandfather used to say. The kind of claim every family seems to carry some version of, had just been checked independently against a molecule by a stranger on another continent whose own grandfather had apparently said something almost identical, in a different language, in a different country, with no way either family could have coordinated the story between them. That's ultimately what this entire body of research amounts to, once the methodology is set aside and the results are allowed to speak plainly. Not a single simple verdict, and not a uniform story imposed on every Jewish community, regardless of its own specific history. But a genuinely rare case, repeatedly tested from a dozen different angles by a dozen different research teams, in which a shared identity carried for 2,000 years, through exile, through dispersal, through radically different host societies on nearly every continent, left a signature that a laboratory, 100 generations later, could still read with real precision. Genetics can't tell David what his family's faith means to him, or what it should mean, or settle anything about identity that was never a genetic question in the first place. That was never what this research was built to answer. What it could tell him is that the story his grandfather told him, filed for years somewhere between memory and myth, was standing on firmer ground than he ever fully realized, right up until the moment a stranger's name appeared in his inbox, carrying, as far as either of them could tell, exactly the same 3,000-year-old thread.